maybe first tell us a little bit about NeuroAge Therapeutics and what the company does.
Yeah, so I'm a physician and a neuroscientist by training, and NeuroAge is a brain aging company. So we have a test that can tell you how old your brain is and your future risk of Alzheimer's more than 30 years ahead of time. Super comprehensive testing includes brain MRI, cognitive testing, blood biomarkers, and genetics. And we combine that into a single score that we call NeuroAge. And your brain aging is about 40% lifestyle. So there's this huge actionable piece that something can do about. And we give people personalized recommendations to slow down their brain aging and prevent dementia in the future.
Cool. Yeah, and we'll dive into how the test works, and we can talk a little bit about my scores. I like this particular test. Before we get into it, I guess I'm just curious, like, what are your thoughts on sirtuin 5 and sirtuins in general and brain aging at this point? I mean, is it a smoking gun? Is it like maybe not? Like where does that sort of body of work stand?
Yeah, so we looked at all the sirtuin candidate SNPs, and so some of that actually I published a paper with Serge Lieber in the lab on Parkinson's and SIRT1 SNPs, and there was signal there. But when I looked at all of the SNPs versus aging, and sirtuins did not pop out as the top hits in that. So I kind of moved towards, some other candidate genes as being more important.
Okay. And then, so that work kind of laid the foundation for the NeuroAge clocks that you're working with now. Is that right?
Yeah. So we were really interested in, it's a therapeutics platform, basically. Can we identify master regulators of brain aging as measured by delta age transcriptome clocks? And so we had this to identify candidate SNPs and genes that control the rate of brain aging. And then we realized, but we could also, there's this huge lifestyle component to how fast people's brains are aging. Can we create something that people can actually use to track their rate of brain aging?
Right. And I think you mentioned this briefly, but your estimates are that about 60% of brain aging is genetic and 40% is lifestyle/environment?
Yes. Yeah, that's what we see. So there's this huge actionable piece.
Right.
So, if you have an ApoE4 allele or even 2, which is what our previous research showed, and you're 5 years younger in your brain aging as measured by our clocks, you're less than average risk. So you've mitigated your genetic risk entirely through lifestyle.
So let's talk about what goes into the brain age clock that you're using now. So there's genetics and transcriptomics. So genetics is pretty straightforward, although I guess a question would be, how many variants are you looking at when you create this genetic risk profile?
Yeah, so we kind of do it in 2 buckets. The genetics is a separate risk score because there's nothing you can do about your genetics. So we just do a one-time score. How at risk are you? The more at risk you are, the more you should worry about your lifestyle, and we should all worry about our lifestyle.
So that's kind of how you think about the way you use the genetic piece. Is it tells you to some extent how important it is to get the other stuff dialed in.
Yeah,
but everyone should get the other stuff dialed in to be optimal. Right.
Right. Exactly. Yeah. And, your brain aging does not just relate to future risk of dementia. It also relates to how well you're functioning now.
Right.
So, people with younger brain ages have better memories, faster reaction speed, better mood. So you're functioning better as a whole with those younger brain ages.
And so in the genetic side, we'll talk about this, and I think we'll leave it behind because, like you said, you can't modify it. But in the genetic side, you got ApoE, and that, I think you said, is about makes up about a third of that risk profile or that risk number, right?
Yeah.
And then how many other factors, ballpark, or maybe the exact number, do you consider to get to the final score?
Yeah, so 40 other factors.
40, okay.
And that's from this Nature Communications paper. So we didn't create the score. We just picked a paper that we thought they did a good job.
Okay.
There's a number of different ways you can create this polygenic risk score. So if you go to one of these big testing companies like Nebula, you might get a range of different answers for how at risk you are, depending on which paper, because some paper use 3 SNPs, some use 40. So we just picked one that we thought was good, and they replicated it. So that was important. They took the first data set. It showed that they could predict risk in a second data set.
Got it.
And then separately, we test for rare forms of Alzheimer's. So 1% to 2% of Alzheimer's cases have early onset. So in your 30s, 40s, or 50s, and you have a dominant mutation that puts you almost guarantees that you're going to get Alzheimer's in your life. So separately, we test for those.
Okay. And what's the percentage of people who have those?
1% to 2%.
1% to 2%. So very small. But and in that case, is there anything that can be done?
Yeah, so lifestyle does push it out in time potentially. So lifestyle is still a good thing to do.
Factor, yeah.
And I think I just wrote a Substack on this that I think is very exciting. Is a study out of WashU where they're looking at people as early as in their 20s that have these rare variants, and they are giving them anti-amyloid drugs as prevention.
Right. Yeah, and we'll talk more about the drugs in a little bit, but we now have at least some pharmaceutical approaches that have some level of effect, right?
Yes.
So, okay. So I think just one comment I'll make is one of the things I've noted is that some of these companies they will give you polygenic risk scores for dementia will often leave out ApoE. And I don't understand why they do that, because it's the most important factor that should be going into this, but just something for people to be aware of. If you take if you get by one of these direct-to-consumer tests and you get a polygenic risk score for your risk of dementia, it may or may not actually include the ApoE genotype. And if it doesn't, it's not a very accurate polygenic risk score.
I think this is like a fluke of the literature. So we're in early days with these companies, but that Nature Communications paper, we actually had to add ApoE back into
So they also took it out.
Yes.
Well, and I think on a research perspective, they probably do that because it dominates everything else, right? But if you don't have it in there, it's just a completely inaccurate assessment of your actual risk.
Right, exactly. Yeah.
Okay. So there's the genetic piece, which I think you report as like a genetic resilience score on the NeuroAge platform. And then you've got the transcriptome, which is a blood-based test. Do you want to take us through what that looks like?
Yeah. So all of my work before the company was really in postmortem brain. And so we were able to tell how old your brain is, your future risk of diseases, how well you're doing with your memory based on transcriptome in brain. But it's not very easy to take a piece of someone's
brain. You probably
brain.
don't have too many customers who want to donate part of their brain.
We're like, here, we'll just cut open your head and get a piece of your brain. Yeah. So we needed some way to translate this into living people. And so in some of the cohorts, we had both blood and brain. And what we're able to do is predict the levels of those brain genes from the blood, from 52 RNA levels of genes in the blood. And some of those are the same genes that are going up and down the brain, and some of them are just predictors of the genes that are going up and down in the brain.
Right, correlated.
Yeah, correlated.
And so when people take the test, they send in a volume of blood, right? Are you getting whole transcriptome data, or are you only focusing on those 50 or so transcripts?
We're doing whole genome RNA-seq.
Okay.
And the reason we're doing that is because the data is very useful for other reasons.
Right. And I mean, as you already mentioned, right, one of the goals here is to gather a large enough data set that you can do future, probably biomarker discovery and drug discovery down the road.
Exactly.
Yeah. So there's genetics, blood-based transcriptome. MRI is another piece that you look at. Let's talk about MRI and what do we know about how MRI can be used to assess brain age, brain function. What are we looking for there, and how does that work?
Yeah, so I think structural MRI is actually the closest thing to what we actually care about, which is whether you've lost neurons or not. So, if you get a disease, it's neuron loss in various areas of the brain. So if the neurons are lost in the hippocampus, that causes Alzheimer's, for instance. And you can measure how many neurons, approximation for how many neurons there are just by the volume of that brain region.
Right.
And even without disease, your brain shrinks with age. It's subtle, but it starts even as early as in your 20s that you lose gray matter. And that's to do with loss of neurons and loss of connections between neurons. So, you don't develop Parkinson's until you've lost 70% of your neurons, but you're losing 10% of your neurons and your substantia nigra per decade before that. And it's just a threshold when you start to get symptoms. So we can measure this. It's a really great predictor of how you're doing with your brain aging and your future risk of disease. And, which area is affected the most will tell you which diseases you're at risk for. So it's a structural MRI, takes about 30 minutes. No radiation or contrast or anything, so they're safe to repeat yearly.
Right.
And it will tell you the volume of your brain and give you a brain age.
And this can be done, when I got mine done, I did a whole body MRI. And so in the process of getting the whole body MRI, they did a dedicated brain MRI, and that can be used for the brain age assessment. And there's some specificity about the way it's done and whether it's going to work in the current algorithm. But beyond that, it should be suitable, right?
Yeah.
And so if people go through your platform, you're able to identify them, get them to a MRI provider who will do the MRI in the way that will be suitable for the analysis.
Yeah, so we're partnered with more than 200 MRI clinics in 5 countries. And so we send people for MRIs that are convenient for them. And I should say we're partnered with a Stanford spinout called BrainKey for this portion.
Right.
So we're in partnership with them.
Right. And I talked about BrainKey on the podcast when I did the episode on my whole body MRI. And so what they do is they'll report back to you volumes for different regions of the brain, as well as predicted brain ages based on those volumes.
Right.
So I guess one of the questions I've got is, it makes perfect sense that, volume is going to be a pretty good proxy for neuron loss and things like that. But then there's the functional component too, right? Which is obviously something that we're interested in. So that really, I think, makes up the fourth piece of what you do. And you've got some functional assessments and how do those work and what are they actually measuring?
Yeah, so we have little games that we call neuro games online. There's 5 of them. So a couple of different ways to measure memory and reaction speed and names and faces, which people often wonder, am I misremembering people's names and faces more than I did when I was younger?
Yeah.
We didn't create these games. We created the software for them, but the games themselves have this long clinical literature behind them for 50 years showing that they change with age and that they are predictors of future risk of neurodegenerative disorders. So you will get a score that tells you how you're doing versus the average person, and the average person your same age and sex. And then you'll get an overall age just based on your game scores. And those are free right now. So you can go to our website and play them.
What's your
give
website?
You an age. So you can go to NeuroAgeTX.com and play the games for free. It'll give you an age. It's more accurate if you also have the MRI and the blood testing as well, but it will give you a ballpark of how you're doing.
Right. Yeah. So I've done it. I've done the NeuroAge test. We haven't actually had a chance to talk about my results, but I'm pretty happy with the results. So you can tell me whether I should be happy or what I should be paying attention to. But my overall score was 44.7. So I'm 54 now, just turned 54.
Yes.
Had to think for a minute because I just had a birthday. My genetic resilience was 79%, which is good. So what is that? And again, that's based on the polygenic risk analysis. So I'm ApoE33. So I'm guessing that would kind of put me at 50%. That's probably the most typical genotype. So I must have other genetic variants that are associated with resistance or resilience.
Yeah, so about 30% of that score is due to ApoE alone. Mm-hmm. So you're 3-3, which is the average genotype, which means that's sort of average risk. But then you also lucked out on some of your other of those 40 SNPs that puts you even more at higher risk.
So that means I really don't have to practice healthy lifestyle, right?
We all want to be our sharpest self.
That's right. And this is, those are that while I might not get dementia, there's other bad stuff that would happen. And certainly looking at my family history, I'm sure that would be the case. MRI put me at 47, and that's based on the BrainKey analysis. And actually, if you go look at their the data they give back to you, they show you each region of the brain and they have numbers for that. You and I were chatting about this a little bit before. I think on the BrainKey, though, that they take into account your chronological age to get that number, right? In other words, it's not an independent measurement. It's based in part on your chronological age and then your delta from that.
Yeah, they're predicting your delta between your chronological age and your bio age, as opposed to predicting the bio age itself.
And then blood, I got 49.7. So that's based on the transcriptome.
Transcriptome. Yeah, so that's your 52 RNA levels of genes.
And does that take into account chronological age, or is that independent of chronological age? Okay.
That we don't use age in any of our internal algorithms.
Okay. And then the games, 22.3.
Yes.
Wow. So
it's like optimal.
Is that something that, I mean, do you have enough data, and is that something that is, I guess what I'm asking is, do most people score low on games, or, because that's way low, like, or is it just some people are predisposed to maybe doing better on the cognitive games?
So it's a great score, and there are some people that we're seeing like this, but I mean, it does have a, good R-squared with age. So, typically someone for your age would be scoring around your age.
Wow. Okay.
But there is some education bias in it.
Sure.
So people with higher levels of education, which your PhD, that's a little bit of an advantage for some of the games.
Right. And again, we sort of touched on this off camera, but there's lots of stuff that's associated with education as well, socioeconomic status, and, more likely to live a healthy lifestyle.
Exactly.
All these things are going to be interconnected. But so I think you gave some numbers, or I've heard you give some numbers on, what it means if your brain age is a certain amount less than your chronological age in terms of Alzheimer's disease risk.
Yeah. So in our study, we showed that people that are 5 years younger, and that is one standard deviation from the mean.
Got it.
This is the equivalent of getting a B in brain aging, so it's obtainable. People can do it. Are 6 times less likely to have Alzheimer's in the future.
That's a huge difference.
It's huge. It's as big as ApoE4 is this aging risk, and it's independent from it. And if you know both your ApoE status and your brain aging score, they create an even better predictor.
Okay. So again, we have to be a little bit careful, right? So what you know is there's this association, right? That across the population, people who have a brain age 5 years younger than their chronological age are 6-fold less likely to develop Alzheimer's disease. The hypothesis is that if you move your brain age in that direction and get to that point, maybe starting from right on your chronological age to 5 years less, that you will reduce your risk by 6-fold. Don't actually know that yet, but it's, I think, a pretty plausible hypothesis. Does that fit kind of with the way you think about it? That's
true. We do have longitudinal data. So people had longitudinal memory in our study, but we only had the one actual brain age measurement, which was at the end. But we were looking at rates of decline. So there is a longitudinal component.
So take me through what you mean by that. What did you look at rates of decline in what?
Cognition and memory.
Okay. So you could see their change in memory, but what you were missing from that study was the same brain age score based on the transcriptome at the earlier time point.
Yes, because we weren't taking pieces of people's brains that were alive.
Yeah, right. Yeah. That might also affect their brain score and cognition. But there is some, so again, the hypothesis is that, people should be able to move their brain age through modifying lifestyle. I think good reason to believe that. And there's some evidence beginning to accumulate in the literature. There's actually quite a big body of evidence in the literature on lifestyle and risk of dementia. And I think we're starting to get some evidence even from clinical trials now. So maybe you can walk us through that. What do we know about the impact that lifestyle factors can have on risk of dementia in people? Yeah,
so, we think it's huge. So similarly, risk of Alzheimer's, unless you have one of these rare forms, is about 60% genetic, 40% environment and lifestyle. And we've seen people in serial MRI with just 25 extra minutes of exercise a week able to increase their brain volume on MRIs. You can literally grow your brain back with better lifestyle. We've also seen there was, this is N of 1, so take it with a grain of salt, but there was a gentleman in Florida who has 2 ApoE4 alleles, so he's very high risk for Alzheimer's in his 50s, and was testing positive for amyloid, which is this toxic protein that builds up, underwent significant lifestyle modification, came back, got retested, was testing negative then for amyloid. So he had removed this risk factor with lifestyle alone. And then a paper came out from Dean Ornish last year showing that with intense lifestyle changes, people were able to slow their early Alzheimer's by 18% in cognitive scores.
So these are people who had presumably mild cognitive impairment, early phase of Alzheimer's, and then they put them in a lifestyle modification program and showed that compared to, did they have a placebo control in that study, or that they could compare it to?
Yeah, it was a randomized control study.
So compared to people who weren't in the program, they were able to delay or slow the progression by 18%.
Yeah.
Okay. So yeah, I mean, so all very, I think, compelling evidence that lifestyle can have an effect. So a couple of things I want to double click on quickly. So you mentioned the amyloid test. So there's a blood test now that will measure amyloid in blood. And there's also the ATN test, which measures amyloid and tau and NFL. Yeah. So, so you're not using those yet, but is that something you're looking at adding into the mix here?
Yes, I want to know my own. I want a baseline, so I'm planned to get the testing. And when we're partnered with clinics, they're often having a baseline.
Right.
We haven't started including it in our testing yet, but it's something that we would definitely consider, because I think it would make an even better predictor. So we would think that information plus the NeuroAge test would improve our results even more.
Yeah, that makes sense. Then of course I can't get through a podcast episode without talking about rapamycin. But
okay.
I did an episode recently, and one of the reasons why I'm really interested in this in the context of brain aging is there were 2 studies, I think, recently that came out in ApoE4 heterozygotes showing short-term treatment with rapamycin increasing hippocampal volume and caudate nucleus volume by MRI, and then also cerebral blood flow. And so obviously there were small studies, but don't want to get ahead of ourselves. But I guess the question would be, how compelling would you find it if an intervention, whether it's lifestyle or drug, longitudinally could show increases in brain volumes in those regions? Like, is that something that you'd be like, oh my God, that's amazing? Or like, that's interesting, but I'd want to see more?
I mean, brain volume, aside from function, is really the bottom line. So I would find that very compelling. I mean, we would want to look at the magnitude and the statistical significance.
Yeah, absolutely. Yeah, my recollection was, because I did a back of the envelope comparison to, I think, the Ornish. No, maybe it wasn't the Ornish study. There was one lifestyle study that looked at brain volume, diet study, and then an exercise study. And it was comparable or bigger with rapamycin, but again, it was a small study. So early, but intriguing. And I would say MRI is great because, I mean, it's expensive. That's the con, but it's non-invasive. Like you said, it's non-radioactive and it's quantitative, right?
Yep.
So these things are measurable and answerable. It's just a matter of getting the data and doing the studies.
Absolutely. Yeah.
So we touched on the drugs briefly. So let's talk about so we know lifestyle is important, and actually maybe before we get to the drugs, what are the key things that you focus on when it comes to lifestyle that can have a positive impact on dementia risk, cognitive function, the parameters that you measure in your NeuroAge score?
So most of it is what you do to take care of your body is taking care of your brain. So there's 6 big ones: exercise, sleeping 7 to 8 hours, diet. Mediterranean diet is good. Community, so social interaction, staying mentally active. Retirement's really bad for that. And lowering stress. So cortisol actually shrinks your neurons. It's bad for you. So anything people can do for time management and, modifying their interpersonal to lower stress.
Right. I mean, those things are obviously interconnected. And, we talk about 4 pillars: eat, move, sleep, connect. And certainly stress is going to be related to connect, but also the other pillars. The one, I guess, that isn't obviously integrated into those 4 pillars is the sort of staying mentally active piece. Yeah. Does it matter what kind of mentally active? Do we know? Like I've heard people say that, crosswords aren't that helpful, but things that require a little bit more, mental flexibility, like learning a new language or an instrument, are going to be better. And do you have thoughts on that, or do we just not know at this point?
Yeah, I think it's anything new that you learn is good. So something that you can do in your sleep is not, even if it's a crossword, is probably not going to be helping you that much.
Right.
But, most people get it through work. So that's why we see this huge drop off around retirement. So if people don't have a hobby that is then
Right.
engaging them, then you lose that piece.
So the message isn't don't retire. It's
Just
when you retire, keep yourself mentally active,
active.
keep learning new things, and
Right.
engage your curiosity. And
Right.
don't just sit on the couch and watch TV. Probably is the way
Exactly.
to say it.
Yeah.
Okay. So now the medications, right? Because I think there's a lot of controversy maybe about the medications, and what is sort of your take on the sort of newer drugs that have been approved or that are coming down the pipeline for treating Alzheimer's disease?
Yeah. So Alzheimer's therapeutics have a controversial history. So when I started medical school, there was a professor at University of Pittsburgh named Bill Klunk who invented Pittsburgh compound B, which is the PET ligand that allows you to see amyloid in the brain. And so what he said, he was very skeptical because he was seeing a bunch of people that were cognitively normal that had lots of amyloid in their brain. And so the question was, okay, if amyloid is the sole cause of Alzheimer's, how could this be? How could there be people with lots of amyloid that don't have Alzheimer's? And so for a long time, I was only working on brain aging. I said amyloid is a red herring. And then there were a bunch of drug failures, with Agilem a few years ago. And I was like, okay, this is total nonsense. We've spent billions of dollars chasing things that don't matter at all. And then we had a couple of wins, which changed my mind. So Eli Lilly and Biogen and Eisai just in the last 2 years each had an anti-amyloid antibody therapy that removes plaques from the brain, does a very good job of that, and they work to some extent. They're not perfect drugs. They, improve memory by about 20 to 30%. They have some severe side effects. So there's what's known as ARIA, which is brain bleeding or swelling in about 20% of people. So you have to have careful monitoring. They're also IV medications. So there's a lot of issues with them, but they are stopping Alzheimer's in its tracks for about 50% of people. And they work better in people earlier on in the course of disease and with some other biomarker profiles that we know about. So I think that's telling us that amyloid does matter. It is a risk factor. I think it's one risk factor amongst many. So this is where my thinking has evolved. So, having high cholesterol doesn't tell you whether or not you have heart disease. And there are people that have high cholesterol and no heart disease. There's people with heart disease without high cholesterol. But it tells you have a modifiable risk factor for heart disease. And I think that's what amyloid is. It's a modifiable risk factor amongst many.
And do we know in the people who are showing... So first of all, I think you said a couple things there. You said for some people, it seems to kind of halt the progression.
Yeah.
Do we know yet, or has it been long enough to know, is that sustainable? Or is it just for some period of time and then they continue to go down the track?
Yeah, so this is 1 year. We're 1 year out this data.
So still pretty early.
It's pretty early to know. But the cool thing, at least about Lilly's drug, is that it's a one-time treatment. So it takes years for amyloid to build up. So maybe you can just take it, get rid of the amyloid, then you don't have to take it again for a decade,
Yeah.
And there's now trials in people as early as 20s, prevention trials, looking at one of these Lilly amyloid drugs to try to prevent Alzheimer's in people that are young people that are very high risk. So they have familial
So these
forms.
are the people who have that 1 to 2% high genetic risk. So they're in their 20s. How long would they be expected to go before they start showing symptoms?
So this trial is they have to be at least 10 years younger than their family members who have gotten Alzheimer's.
And are they going to wait that whole 10 years before they read out the results?
Yes.
Wow. That's more power to them, but that's pretty rare that a company is going to do a 10-year clinical trial. So that'll be really interesting.
Yeah.
Unfortunately, we'll have to wait a decade, but we'll keep our fingers crossed. And I think like you said, there's nothing saying that you have to only do one, right? So maybe the amyloid drugs combined with lifestyle, combined with other things that are going to be developed in the interim.
Yes, and there are safer forms of those drugs coming down the pipeline.
Sure.
So small molecule pill forms that don't have ARIA, they don't have the side effects. And then there's also other toxic proteins, so tau. There's also tau drugs. So some people might do better with amyloid drugs, some with tau drugs. And then another thing that I think is really exciting is actually the GLP-1 drugs.
Oh, yeah. Let's talk about that. Yeah. GLP-1 agonists. Yeah.
Yeah. So there is, so they looked at people with diabetes that were on a GLP-1 drug versus another diabetes drug. So presumably that's somewhat controlling for how well their diabetes is being controlled.
Right.
And what they showed is that people on the GLP-1 drugs had less incidence of dementia.
And what's the relationship between diabetes and dementia? That you have an increased risk if you're diabetic, and that's how big is that increase in risk? Do you remember?
I don't know the exact numbers on that, but it's significant.
Yeah, it's I kind of feel like it's 2 to 3-fold, but I don't remember the exact numbers. So, I mean, the reason I bring that up is, you always wonder, would GLP-1 drugs have an impact in people who are non-diabetic, right?
Exactly.
We don't know the answer, obviously, but
and I did a little bit of a deep dive on that, and so there's another question, which is if your glucose is not diabetic levels, but is still not ideal,
like so pre-diabetic,
or even the ideal glucose range is in your 90s, which is well below what would be considered even pre-diabetic. And so when you look at older people with better glucose control, even if they're not diabetic, are showing less dementia. So should it be in people that, should we be taking these in people that are not even pre-diabetic just for good glucose control?
What are your thoughts? Because this is something I'm super interested in on the trying to separate the effects of the GLP-1 agonists on caloric consumption and body weight from, potentially other effects, right? Because most people who take GLP-1 agonists, even if it's for diabetes, are going to take doses that suppress appetite. So they're going to eat less. They're probably going to lose weight. That could have effects. Have you seen data to suggest that there may be effects that are independent of appetite suppression and weight loss?
Yeah, so in some of those studies, they controlled for weight loss, right? So it seemed to be separate from that. And the cool thing is I did a little digging into the mechanism because I was like, how could this be working?
Yeah.
And they're working through PI3 kinase, which is AGE1, which is a known longevity pathway. And what it was shown, at least in animals, is that they're protecting cell death in both the pancreas and in the brain.
Right. Because the receptor is expressed in the brain as well, right? Yeah. Which is super interesting.
Yeah. So they may be, I think that this is going to end up being, a longevity drug, really, was the way we're going to be thinking.
Yeah, I'm intrigued and I'm bullish, but I'm not yet ready to
say that.
Yeah, I mean, I'd love to see the experiment, right? I think that's, I mean, I think they would be longevity drugs, at least in mice, because they would also cause caloric restriction. But I think if you were to control for that, there's a plausible mechanism by which they could also have effects on longevity independent of caloric restriction, which is really interesting. And so, it'd be really cool to see how this plays out.
Totally agree. And then the other thing that I would highlight is HRT. So in women, so hormone replacement therapy. So there's some good studies coming out showing that women that have hormone replacement plus minus 10 years of menopause, that's really important.
And so what you mean by that is hormone replacement after you've been in menopause for 10 years, you might get different effects than if you started within 10 years of
so there was a study that came out from the Women's Health Initiative in the early 2000s that scared a bunch of women off from taking hormone replacement therapy, but that was done in women 65 plus.
Right.
And so when they redid that study in people that were taking it younger, what they showed is that it's detrimental if you all of a sudden start taking hormone replacement therapy when you've been menopausal for 10 years.
That was mostly a tiny, tiny cancer risk, right? I guess what I'm wondering is, did they even look at cognitive function and dementia in that study?
They did. There was a slight increase in risk if you started it in
later. Okay.
And the other thing is it wasn't bioidentical.
Right.
So that it was like this synthetic HRT that most people are not prescribing at this point from horse urine.
Because of course.
Yeah. So if you're, most women go through menopause at 50. So if you're between 40 and 60, I think, and you're worried about Alzheimer's in the future, I think it makes a lot of sense.
And do we know what the magnitude of effect looks like for HRT in women in terms of dementia risk?
I don't have that figure off the top of my head.
Okay. Yeah, I always just find it interesting to kind of think about, we know what the genetic effect size is. We know kind of what the lifestyle effect size is, just to kind of see how big are these levers. But I'm guessing it's probably comparable to lifestyle, but we should get that, and we can link to some of the studies and put it in the show notes.
Yeah. And I can tell you, for me personally, I started HRT about a year ago. I'm 43, and I was having in my late 30s, early 40s, insomnia. So I was waking up like 5 times a week at 3 in the morning, having a hard time getting back to sleep. It was making me exhausted during the day. And I thought, oh, this has got to be stress. I'm an entrepreneur,
that'll do it.
And then I started HRT like almost a year ago, and it fixed my sleep. Now I wake up twice a month at 3 in the morning. It's amazing.
It is amazing, and I think this is a good example where you're younger, I think, than most people would think about it. But I think for some people, like, it makes sense to go get the blood work, see where you're at, find a good doc, and it can have big effects. So don't wait, I guess, would be the question to find out, right?
And if you look at the estrogen curves, it's your late 30s, early 40s that you start to see them falling off the curve. So perimenopause often starts in people's early 40s, late 30s.
Right. Yeah. Great. Well, this has been fascinating. I want to just shift gears a little bit to the extent that you want to talk about it, because I know, you've kind of pivoted a little bit in your business plan. I guess I didn't say this, so I should say, at full transparency, I'm on the scientific advisory board of NeuroAge. But you kind of made a shift a little bit towards a consumer-based model. And you and I, again, we're chatting about this a little bit off camera. And as anybody who watches the podcast will know, I'm pretty skeptical of the direct-to-consumer biological age tests in general, which I guess you would call that. So we should talk about that. Like, what is biological age and what does it mean? But in particular, the epigenetic age tests I'm skeptical of, because I just some of my own data that I've seen and talking to lots of people. So I guess I'm just wondering, kind of, how do you think about that? How do you navigate that landscape of choosing that you're going to market a product to consumers? You also have clinics that you're working with, and trying to balance accuracy, giving people, and I know you have the right intentions. You want to help people for sure. How do you balance that with the business model, right? And you got to make a profit at some point. So
right. So I think you and I think about the clocks a little. I'm a little bit more bullish on them than you. So I'll just say, like, I sent my blood off in duplicate to Clock Foundation, got 6 biological age tests back. I think everyone should do it if you're willing to sort of sift through what that means. So I got GrimAge and PhenoAge and Horvath and so they came back ages 22 to 55 about.
So let's double click on that and clarify what you mean. So you sent your blood to the Clock Foundation, which is a nonprofit. And they measured your methylation status and ran 6 different algorithms on that, which are different metrics that are in the scientific literature for measuring epigenetic age, right?
Yeah.
Okay.
And I didn't tell them how old I was, and I sent it in duplicates.
And what were the scores you got back?
It's 22 to 55 about.
Okay. Okay.
So I'm 43. My average was below my chronological age, which made me happy. If you just take a mean, my PhenoAge was really good.
Yeah.
And my GrimAge was good, but the second generation, or the Horvath clocks, like the older ones, were the older ages. Anyway, so, but the duplicates were coming back within like a year or so of each other. Mm-hmm. So that was reassuring that at least the technical replicates are
that the Clock Foundation
is doing.
Uses. Yeah.
Yeah. So, but, I think they're measuring different things. So, like, PhenoAge it depends how you define biological age. So I don't think there's one biological age. I think there's different ages depending on organ and how you define it. So PhenoAge is taking into account, like, it's trained on whether or not you have diabetes, whether or not you have heart disease. The pheno part is, all of these different disease risks. And so it's going to tell you more something about your future disease risk. Whereas GrimAge is really, and Dunedin Pace, right, are, or Dunedin Pace, I think.
Yeah.
Dunedin Pace.
Yeah.
So they are more telling you about lifespan, or pace of aging. So the way those clocks were trained is different. They're training on like how old do you ultimately live, which isn't just your disease risk. So you're going to get different
and
answers.
also trained on different populations.
Different populations.
Right? Different, because I mean, all of these, as far as I know, have been trained off of biobanked samples from different longitudinal studies, right? So I agree with everything you've said. The algorithms are different. They were trained differently. They're measuring different things. They're all somewhat correlated with health or mortality risk. I guess I have two comments. One is, why do we call them all biological age then if they're all different? They can't all be measuring biological age.
Yeah. I mean, there's a nomenclature problem.
There is a huge nomenclature problem.
Yeah.
And which one's right?
So I don't think there's a right answer. I think they're giving you answers about different things.
Okay, which one's useful?
Which one's useful? So for me personally, what I'm doing is just taking the average of them.
Okay, but that's not very scientific.
Yeah, it's not. But I mean, you can pick one and track it longitudinally and say, okay, I'm going to go with this clock. If I had to pick one, it would probably be one of the newer ones, like PhenoAge or Dunedin Pace, if I'm going to pick one.
Just because it's newer, or because it...
Well, they're a little bit better. The problem with the original Horvath clocks is that they were really accurate. They could predict how old your birthday age is by like within R squared, like 0.99.
Right, chronological age. Yeah.
But like that noise is the real noise. Like that's really what you care about. Like it's not just a worse predictor. You know, this is what biological aging is. So if you remove all of that variation, then you're only predicting someone's birthday age.
Yeah, I guess I would still question whether that's really biological aging, though, right? I don't know the answer, but it's one, in this case, one biological parameter, which is methylation, right, that changes with age.
Yeah.
Is that biological aging?
Well, think about how you can, like, we have really good algorithms. You can look at someone and more or less tell how old they are.
Just by looking at them.
Looking at them. Yeah. And there is face age as one of these new clocks. But that's a combination of what? Like muscle tone, wrinkles, gray hair, balding, even voice quality. Like all of these things combined together to give you a picture.
Right, which we as animals have evolved to sort of interpret, right?
Yes.
Yeah.
Yeah. So probably the most accurate one would be the one with all of these measurements, right?
I guess that's part of my, I mean, so there's the question, I think, which I think is a bigger question, which the field is struggling with. And as you said correctly, there's a nomenclature problem that different people are using the term biological age to mean different things. And yet nobody's actually communicating with each other. But then I think the question also is, what is the most useful things to measure then? Is it, like you were saying, the sort of integrated piece? Is it the functional piece, like how well your heart's working, or, how much weight you can lift? I think that's a reasonable thing to look at. Or is it the molecular markers? All different types of information, and I struggle with which is most useful or informative or actionable.
Yeah.
But I think the other piece, which is a different question, is the direct to consumer side, right? So like, I think you and I both agree on the research side, these are very interesting, powerful tools. Someday will tell us mechanistically about the biology of aging. Someday, hopefully, will be useful in clinical trials, like all of that. Yeah. I don't think we're there yet. I'd be interested if you agree or disagree on that piece.
I mean, I think that, so I think I would love if every clinical trial ran 12 clocks.
Just to see what works.
See what's moving with drugs.
Right.
See what's the best predictor. And I think, if you had a GLP-1 trial and 10 out of 12 clocks moved with GLP-1s, I would be pretty convinced by that as an additional piece of evidence.
Yeah.
So I do think there's some utility now, but there is a lot of things left to work out.
I agree completely, and I also think the fact that you suggested we need to do that tells me you don't really believe the clocks work yet.
not that I don't think they work. It's that I think we need to figure out what is the most useful one of
which ones
which ones for which purpose.
Okay.
Yeah.
And then I think the other piece, though, is the direct-to-consumer side, right? So,
but I will just interject really quick. I would agree with you to some extent. That's part of why NeuroAge has all these pieces.
Yeah.
Because I said, what would I believe?
Yeah.
What would I believe? I'm trying to track my own rate of brain aging for my own dementia prevention.
Right.
And for me, it was like, well, if you show me the convergence of brain MRI and cognition and, the blood testing, and you combine that together, now this is a predictor that I have faith in.
100% agree with you. And I think that, I'm glad you said that, because it is worth differentiating a kit that you get in the mail and you send off and they send you a number back with your methylation status, right?
Yeah.
I think what you're doing, the fact that you've got MRI, which is quantitative, we know brain volume's important, right? Yep. You've got the games that people play, reaction time, word pattern matching. Again, we know that's important. We know it's quantitative. You can measure it. The RNA piece, I guess, kind of falls in the same bucket as the direct-to-consumer epigenetic test, but you're not only relying on that, right?
Yes.
And then the genetics. Again, you're giving people actual information on their specific genotype, which we know, especially for things like ApoE, are real. Like, it has a real impact, right?
Yes.
So it's less, it's more direct and quantitative, and I have infinitely more confidence in those things than I do in a epigenetic test right now. Again, not because the epigenetic changes with age aren't real. They are, and you can measure it in the laboratory and you can correlate it with health outcomes and mortality. But I think the piece that's missing for me there is the mechanistic connection. We don't yet know which epigenetic changes are causing, which gene expression changes are causing which diseases or mortality risks, right? Nobody's made that connection yet.
Which is part of the experimental part of what we're doing, right? Which is that we're tracking both methylation and transcriptome. We're not giving people methylation results.
Right. But you'll be able to, down the road, figure out
it's
what
part
the
of our
connection
clinical
is.
study.
Yeah. Great.
So, if you connect multi-omics, we also want proteomics and metabolomics.
Right.
And if you put that all, now you've got a picture of what's going on that makes sense, has real biology underpinning it. It's not a black box.
Right.
You know what's going on.
Right. Great. So again, I don't want to spend too much time bashing the direct-to-consumer. Well, actually, I do want to do that because I think it's important that people actually understand.
Mm-hmm.
But again, on the direct-to-consumer side, though, like, you went to the Clock Foundation. They're a nonprofit. You're right. Nonprofits can make money too, but they're not at least beholden to exactly the same incentive structures that a for-profit company is. And I guess I'd be interested to know your feelings on one of my concerns, which is that there's really no transparency into the quality control of the tests themselves, right? So again, I did 4 different companies in duplicate. I think 2 of the companies, the duplicates came back close together. 2 of them, they were, like a decade apart or 2 decades apart in one case. And they were all over the place. So I got all the way from, like, I think 43 or 45 to 67.
Mm-hmm.
For my ages.
Yeah.
So it could be that the companies are using different algorithms. It could be that the whole industry is just a mess, which is sort of where I land right now. But my concern is the consumers don't have any understanding, even if it is different algorithms, why they're getting different numbers from the same company when they do the test longitudinally, or from different companies when they do it across different companies.
Yeah. Well, there's certainly also this we need to be better explainers, which you're doing with this podcast, of what this stuff actually means. Because I think people are they get one test, tells them they're 5 years older, and then they're freaking out.
With that? Yeah.
And they're very upset, know, and of course you're upset, Like, well, I saw in MIT Tech, there was an article a couple of years ago where one of the writers took a test and said she was older, even though she's, like, very fit and does all this yoga and stuff. And it was upsetting. Right. I get it. So I think people just have to be aware of all of the caveats with where we are, because it's very early days in terms of this testing.
So let me ask you, maybe you don't have the answer yet, but one of the things that has also bothered me about the epigenetic tests is I've got longitudinal measurements over couple of years now, right? Let's just say every 3 months. It wasn't quite that frequent, but ballpark. And the numbers kind of go like this, which is not going to surprise anyone. And then they measure the Dunedin pace, or the pace of aging, which is supposed to be your rate of aging.
Yeah.
And the rate of aging doesn't match up at all with the numbers that go like this.
Oh, really?
And in fact, in one case, like my rate of aging would have had to been 900 times the normal rate of aging, which is silly. So I guess the question would be, again, how, first of all, the question I wanted to ask you is, with your NeuroAge test, do you know if it is robust against that sort of wild fluctuation over time in the same individual?
Yeah. So we are early days, so we've just launched the test. But from what we know from the postmortem data, we have reason to believe that it's going to be stable, especially brain MRI. I mean, it's kind of hard to... but there is going to be day-to-day variation, like in the cognitive testing. So if you get a better night's sleep, you're going to have better reaction time.
Yeah, exactly. The games. Yeah.
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 So, and that's, I think, a feature, not a bug. That's sort of like, there's this, how you're doing, are you sick that week? It's going to make your cognition worse, or not sleeping as well. But then it's going to average out to, where your average cognition, if you do it a bunch of times.
Right. I think it'll be interesting to see over time how your algorithm performs in terms of that variation. I'm not even sure that the variation is wrong.
No.
I mean, it might be telling us something fundamental about the biology of aging we didn't previously appreciate. But also, and you alluded to this, I think it depends on what you measure, because some things will be resistant to that sort of variation, like brain MRI. I would guess you're not seeing huge changes in brain volume in a circadian cycle, right?
No.
Although I don't know. I guess we would need to do, 24-hour MRIs maybe, but I'm guessing it's not a big effect, right?
Yeah, it's not a big effect. And with I should say with our RNA testing, we have done like 1 week apart duplicates morning, evening, and showed that we are not seeing
relatively stable. Yeah. I guess the other piece though that I wonder about, and I'm actually leaving for Singapore in a few days to go to this gerophysics meeting, and I know this will be a topic of conversation there, is, maybe there's really a couple of dimensions, right? There's the age-related change, which we all think of as sort of an exponential, I guess. And then within that, there's a variation, right? And different tests are going to measure different aspects of those two dimensions. And again, there's probably more than two dimensions, but
yeah. I mean, if you just think about glucose, right?
Yeah.
You may end up with, sometimes you're having big spikes and sometimes you're more stable.
Right.
If you get a cold, you have worse glucose.
Right.
If you sleep worse, you have worse glucose variability. So,
and then as you get older, right, your resilience to that variability presumably is going to decline.
Yeah.
Or you're more likely to get pushed over the threshold to pathology or mortality, right?
Yeah.
Potentially.
And the same thing with inflammation. So I think of it as like us oscillating superimposed on something that's going up.
Exactly. Yeah. So there's lots to learn, obviously, but I think, what you're doing is fantastic and going in the right direction. And I'm really excited to see what happens next. So maybe you can tell us a little bit about your background and how you even got on this path and what got you interested in brain aging and brain health.
Sure. So I have a family history of Alzheimer's disease. So when I was a kid, my grandmother suffered with Alzheimer's. My grandfather took care of her in the home. It was about a 7-year disease journey, had a really big impact on me as a child. Now, and through the course of NeuroAge, I found out that I myself have a genetic risk factor for Alzheimer's disease. So I have an ApoE4 allele, which puts me at about 2 to 3 times the risk as the average person.
Right. So you have one copy, right?
I have
You're
one
carrier?
copy.
Yeah.
I'm a carrier.
And what's the percentage again in the population of people who have one copy of ApoE4?
So about 20% of people.
Yeah, so it's a pretty big fraction, yeah.
It's super common, and people that have 2 copies, about 2% of the population.
Right. And that leads to a much higher, like 10 to 15-fold increased risk?
Yeah, if you have 2 copies, it's more than twice as risky as one.
Yeah.
Yep. And so that led me to dedicate my life to coming up with better solutions for Alzheimer's disease. So, at our heart of the company, we're a therapeutics company. We think of ourselves like Tesla. So Tesla is selling cars in order to gather the data for self-driving. We are selling the NeuroAge test to gather the data in a privacy-protected way for better Alzheimer's therapeutics and biomarkers, because we really need a lot more data in order to have good solutions for people. And in terms of my career trajectory, I started off on a traditional path. If you ask someone, how do you create better drugs for Alzheimer's, they say, well, go do an MD PhD program. So that's what I did. I completed my MD and my PhD in neuroscience at University of Pittsburgh School of Medicine combined degree program. And then at that time, I thought I was going to be a neurologist. But there wasn't a whole lot we could do for people with Alzheimer's. There still really isn't, although there's more we can do now than we could when I was in med school.
Yeah.
And so I instead decided to purely focus on my research, and I went to MIT to then pursue a postdoc. I started NeuroAge a couple of years ago in 2022.
And that was based on your PhD research to begin with, right? A lot of the background data for how you're approaching this at NeuroAge. Let's talk a little bit about that and kind of what you learned from that process and how it set the stage for what you're doing now.
Yeah, so when I started graduate school, so I'm going to date myself, this is 2003, I really wanted to be in a lab that had unbiased approaches, so that was one thing, and had human data. So I really believe that what we were doing for neuro in particular was a lot of serendipity, was a lot of drugs that, were found by accident and kind of work. They didn't, especially in neurodegeneration and also in psychiatric disorders. So I joined a lab in University of Pittsburgh psychiatry department, which has one of the oldest human brain banks and most extensive human brain banks, and started working on what happens during normal brain aging that contributes to disease. And at that time, a lot of people were very focused on the amyloid hypothesis for Alzheimer's, which I can get into. It's these toxic proteins that build up in the brain. And there wasn't a lot of research looking at what happens during normal aging, even though age is the biggest risk factor for Alzheimer's disease. So I published a bunch of papers, some of them on mice. We actually started looking at, so that lab had what was, I think, the first aging clock. So my professor, Etienne Seville, during his postdoc at Columbia, had created a transcriptomic aging clock in human postmortem brain. That work was published in 2004, so nearly a decade before the Horvath clocks came out.
Wow.
Yeah, and so when he showed me that data, I was really impressed by it.
And so basically, when you say transcriptomic clock, this is going to be brains that were donated looking at RNA levels of different genes and then finding a subset of genes that are differentially expressed that can predict chronological age in this case?
Yes. So he was a postdoc in Renee Hin's lab, is a famous mouse lab at Columbia, and they were looking at depressed brains versus control. They said, can we see what's different about someone with depression than someone who doesn't have depression?
Okay.
And they didn't see much. Wasn't a big N, at that time they had, 20 or so brains they were looking at control versus depression.
Right.
And so then they said, can we see any differences? So let's look at sex and age just to see if we can see differences.
Right.
Also didn't see much with sex. It turns out there isn't a whole lot of difference on a transcriptome level between a male and female brain. So
who would think that?
So anyone says there's these huge differences between men and women, how they think, not true, at least at the RNA level.
Well, something's different, but we'll get, that's a different episode.
Different episode. But what they did see were these giant differences with age. It was this huge effect size. And so he had the idea of, can we actually predict how old someone's brain is just using transcriptome levels of genes? And it turns out that you can. And so he was also working in mouse models, so so there was some work on that. And then my work, we said, okay, well, if we can see how old a brain is, are there genetic factors that we can identify that relate to that difference in how old or young someone's brain is? And that brings me to sirtuins, because we had a serotonin knockout mouse paper that one of the biggest genes that was transcripts that was difference between the serotonin knockout and control was SIR2 and 5. And so I looked at SNPs during my PhD and the SIR2 and 5 gene that were related to older or younger looking brains. And so we published that work. And then that got me interested in sirtuins. And so I approached Lenny Grante, who's our common scientific ancestor at MIT, to join the lab. And we continued to work on that. And then I said, well, maybe we shouldn't just look at candidate SNPs. We should look at all the SNPs.
Right.
And also it's really important, not just to be measuring chronological age, but to know that what we're measuring matters for disease. So we looked at longitudinal data with people with Alzheimer's versus control to really tie our clocks to function and disease risk.
Yeah. Is there anything we haven't talked about that you wanna mention?
No, I would just say knowledge is power. Knowing your brain age, knowing how you're doing, is really empowering to improving your tomorrow. And so I would encourage everyone not to be scared of dementia risk, but instead to take that bull by the horns and start working on it.
Fantastic. I think that's a great message to leave with. So thank you so much for coming on the podcast. It's been a pleasure, and I'm excited to see what happens next.
Yeah, thanks so much, Matt. It's been really great.
Awesome.
Appreciate it.